Stayed here in Spring Arbor, so we know the acronym for that one. SAL, right? Okay, so without further ado, these three students who have done research experiences for undergraduate students are going to talk about the fantastic things that they did, and hopefully you'll be able to hear them. So project and rest the mic on your chin. You know, it feels weird.
All right, can everybody hear me okay?
Yes.
All right, so this is a really long title. I didn't exactly come up with it, but the most important word is Amino Mutases, because that is the class of enzymes that I worked with this summer for my projects. And then you see my mentor, his name is Tyler Walter, and the lab I was in was directed by Dr. Kevin D. Walker, principal investigator, PI. It's pretty much synonymous with the director of the lab. So he's the head honcho. And like I said earlier, Michigan State University program is plant genomics. Pictured here on the left, you see my mentor Tyler, and me in the middle, and on the right is a guy named Prakash, who's a fellow graduate student in the lab. I'd like to note that what he's wearing, his sandals, are not considered proper lab attire, yet he insisted upon wearing them pretty much every day to work anyway. So today I'm going to talk about my RU experience, the research I did with enzymes, and then the future of the research / how the experience at Michigan State affected me and developed my future. So plant genomics, it's an RU at Michigan State. It's about 10 weeks, and you work 40 to 50 hours per week, which is like a normal work week if you graduate and you get a full-time job. They take care of you pretty well. You don't have to worry about finding a place to sleep or food. One thing that really stood out to me as a beneficial part of this program was weekly seminars. So our whole program would eat lunch together, and then a PI or a director of a lab would come in and give us a clear, understandable presentation at our level of what their research was and what they were doing. And for me, that was really great because it gave me a clear picture of what kind of things are going on at MSU and what kind of research is out there. And I felt that I got exposed to a lot of different biology-related research that was really interesting. Also, just having a graduate student mentor was an incredible experience to learn from somebody as they're learning as well. And at the end of this, I gave a poster presentation at a conference as well as an oral presentation in front of my program. So that's just an overview of the plant genomics program, and then RU just stands for Research Experience for Undergraduates. If you get a chance to participate in one during the summer, I highly recommend it. It was a wonderful experience. All right. So amino mutases, like I said, they're enzymes. For the purpose of today, I'm defining them as proteins that serve to catalyze chemical reactions. And what I have pictured here is this kind of Lego building block pathway where you start with these two Lego pieces there, and you go through a series of enzymes that catalyze the conversion all the way to this biologically active compound. So that's kind of an overview of what a pathway may look like. Amino mutases themselves catalyze this conversion from alpha amino acids to beta amino acids. Don't get overwhelmed too much with the organic chemistry symbols and whatnot if you're a first year science student. But the important thing to note is this amino group right here, which organic students probably are learning functional groups or have learned them by now. It gets moved from the alpha carbon to the beta carbon here, and that's what amino mutases do. And then these beta amino acids serve as Lego building blocks on the biological pathway towards these active compounds. Here, I've just illustrated this using that Lego analogy. Like I said before, you start with an alpha amino acid. It goes through the amino mutase, which is often the first step in this pathway. Then you get the beta amino acid, followed by going through a series of several enzymes to get to your biologically active compound. So that's kind of an overview of the pathway. Paclitaxel or Taxol, which is it's a large compound. Again, that's the end overall product of these pathways. And it's an example of a biologically active compound that starts with an amino mutase converting an alpha amino acid to a beta amino acid, and then that beta amino acid building block leading up to this final end compound. And what this is a defense metabolite found in specific yew trees that we isolated and then were able to develop as a medicine. Like I said, it's used in chemotherapy. Often it's very effective in breast cancer. And what happened is when they discovered it, worked so well that they over-harvested the Pacific yew tree. And so a lot of what my lab focuses on is using these enzymes involved in the pathway and trying to improve their efficiency, make them more green, make them more effective, and develop a better method for making Taxol so that it relieves stress on the Pacific yew tree. Here, again, don't get overwhelmed with this huge structure, but it's another end product. It's an antibiotic called Andromid, and this relates specifically to the enzyme that I worked with called PA-PAM. It's just an amino mutase that catalyzes the first step in the synthesis of Andromid. The other enzyme that I worked with is called OS-TAM, and it's found in Japanese rice. And the story behind this is kind of cool. Dr. Georgiander at Cornell University found an overproduction of beta-tyrosine in the Japanese rice. Then he isolated the gene encoding for the enzyme and cloned it into tobacco plants. From there, he got the tobacco plants and harvested the enzyme and sent it to us to analyze how the enzyme works, the stereochemistry and mechanism of that conversion. And he and his team are working on trying to figure out why it's producing the beta-tyrosine and what potential medicine you could develop from defense-induced metabolite. So this table kind of outlines some previously studied amino mutases, and it shows the stereochemistry of the beta amino acid along with if it undergoes retention or inversion of configuration. And these bottom two are the two enzymes that I worked with primarily, mutant of the PA-PAM and then OS-TAM from rice. I won't get into the details of how we found out the mechanism and the retention or inversion of configuration, but we used some deuterium-labeled substrates in the chiral auxiliary, if those of you are familiar. And we determined that the product was an R stereochemistry and that we had a retention at the beta carbon. So that was really cool. I helped my mentor a lot with that project. Some struggles of this were that the enzyme was really slow, and often we had to wait for Cornell to ship us the enzyme when we used up what we had. So that's that. And then this is the other enzyme that I worked with, PA-PAM. Here you see the active site of the enzyme pictured with the arginine here, take note of that, and then the substrate L-phenylalanine. And this is what it looks like in the wild type. And then we made a mutation. Here you see lysine is in place of the arginine, and there's a weaker interaction between the lysine and the substrate L-phenylalanine, which produces a cinnamate intermediate. It doesn't take it all the way through the conversion to the beta amino acid. The first step of working with these enzymes was expressing and purifying it. I had a long list step by step in my formal presentation at the end of my program, but I decided to cut that out just to save time. And just the point is that I expressed and purified the enzyme, and this SES page gel is just a way of determining the purity of the enzyme to make sure that what we're working with is good and it's ready to move on to the enzyme assays that I did. Then I developed this reaction progress curve for the enzyme. So as the course of time, it develops a certain amount of product. From there, I developed a Michaelis-Matin kinetics curve. Again, I'm not get into the huge nitty-gritty details. What you might be able to see below is the Km values, but actually KCAT is what I'm going to be talking about in the next slide. So the point is I got kinetic constants out of this. And what we wanted to do is to find out what these Ka and Kb kinetic values are. So we know KCAT from the Michaelis-Matin kinetics curve, but we don't know Ka or Kb. And what we wanted to do was find out Ka, because if we can find out what Ka is, then we can use this equation to solve for Kb pretty easily. And what was proposed was to use phenylalanine now as a like surrogate substrate or just a different substrate to inhibit the enzyme. And as it forms this chromophore, it loses the amine group, and that would be the Ka rate. And we were hoping to be able to track it. Another problem or rate limiting step in this research was that phenylalanine had not been synthesized yet when I arrived. So we were also waiting on Cornell to ship us the enzyme. So I was stuck at a point where I didn't have anything that I could do because the enzyme from Cornell wasn't there, and we didn't have phenylalanine ready to use in this in these assays. So I came up with the idea to use cinnamaldehyde as a different inhibitor. You'll notice it starts with a double bond here instead of having an amino group there, but it yields the same imine chromophore, is what we hypothesized. And so this would not be useful in determining Ka because we're not losing an amine, but what it is useful is determining, will this methodology even work to begin with? Are we going to form a chromophore potentially? So I investigated that, and you see here just the top row is photos with camera flash on, and the bottom is photos with the camera flash off. This is a time course where I incubated the enzyme with various chemicals. The second test tube from the left is the experimental, and then you have to compare to several controls according to this key. But you'll notice the second test tube, it changes color throughout the course of the reaction to this orangish color, which was very exciting as a preliminary result. And we attempted to get UV-Vis data and struggled with it for a while, and we have it now, but at the time when I finished, we weren't able to get it, so I don't have it included in this presentation, but we found out the UV-Vis spectrum to see where this is absorbing. Then we synthesized this hypothesized imine chromophore organically, so we just took, no enzyme, just started with lysine, which is what should be in the active site interacting with cinnamaldehyde, and we synthesized this chromophore. NMR analyses confirmed that this is what we had, and you'll notice the same kind of orange color as what the enzymatic amine was, which is what was really exciting for us, which leads us to believe that we will form a chromophore when we use phenylalanine now. And that's essentially the end of that kinetics research with the phenylalanine amino mutase. But just to give you a kind of cartoon picture of what we did, if you imagine Pac-Man is our mutant enzyme, then you have this yellow dot is the natural substrate, phenylalanine, and the red dot is what we put in, cinnamaldehyde as an inhibitor. When Pac-Man gets to the red dot, the inhibitor binds to the lysine mutation in the active site, and it becomes this orange chromophore. So the future of this is to further develop the enzyme and substrate to hopefully produce a fluorescent tag instead of just a colorimetric tag. And then one idea that I thought would be really cool, and it's really kind of far out or in the idea stage, would be to maybe link this colored or fluorescent tagged enzyme to another enzyme that's, say, hungry for lactate, and then cancer cells, and colon cancer cells specifically, convert the high amounts of pyruvate into lactate, which is produced by the Warburg effects to avoid cell death. And so hopefully what you might see, and this is just an idea, is that you have the fluorescence gets dragged to where the colon cancer cells, and it's a way to identify where the cancer exactly is in your body. So that's just an idea that might be able to be produced way down the road from this research. So just to recap, we looked at the OS10 enzyme and what the mechanism and stereochemistry was. I developed Michaelis-Matin kinetics for the mutant enzyme of PACAM, and we looked at cinnamaldehyde as an inhibitor for this enzyme. So that's the research recap. Struggles with this, like I said, phenylalanine hadn't been synthesized yet, so we couldn't use that. OSTAM is a slow enzyme, and we had to wait for Cornell to ship the enzyme to us. And then the Diazo Methane, this was the biggest of them all. It was faulty or it was too dilute. Whichever way, it was really throwing off the results of our enzyme assays. So for weeks I was collecting this data that just did not make any sense. And when we finally figured out that our Diazo Methane was off and we swapped to a different bottle of Diazo Methane, the results started coming out just perfect. And like you saw the reaction progress curve and that Michaelis-Matin Kinetics plot that I had earlier, it was a nice smooth curve. That was developed pretty much the last week that I was in the program. I probably did, so biochem students, you recently did an enzyme assay, I believe, for one of your labs. And I probably did 150 assays in that last week that I was there to try and collect that data once we figured out what the problem was. So I worked really hard to develop it, and those were some struggles with the research. My future, before this program, I saw myself as wanting to go to graduate school and then maybe become a professor and just teach. But I really found that I thrive in a research lab environment, and really, I just loved it. Loved being able to be on my feet working and doing reactions, and every day brought something new. And I just loved the challenge of being in lab. So after the experience, I could see myself anywhere from just doing straight research, all the way to a combination of teaching and research to just teaching, because I have that broader interest now. Also, before, I was very focused on chemistry and my love for chemistry, and while I still love chemistry, my exposure to biology and knowledge of it increased, and I found that I really enjoy that as well. And graduate school is for me, and that's my future direction. I'd like to thank the Walker Lab, my mentor, the Plant Genomics program, the National Science Foundation for funding, Dr. Georgiander at Cornell, and the SAU Science faculty for supporting me throughout this experience. And thank you. Are there any questions?
Let's save questions.
Okay, we'll save questions to the end.
Yeah.
All right, so when I was in Texas, I studied the cross-reactivity of 4 novel monoclonal antibodies against the secretory glycoprotein of the Ebola virus. And this was in the ROX lab in the Galveston National Laboratory. So just some quick general facts about Ebola. Ebola is a non-segmented, enveloped, negative strand RNA virus. And basically that means that the RNA in its genome can't immediately encode for messenger RNAs. It has to be converted into positive stranded RNA via RNA polymerase. And its main targets in the body are endothelial cells, macrophages, monocytes, which are another type of white blood cell, and hepatocytes. The endothelial cells are particularly disconcerting. That's what causes hemorrhagic fever. And that's part of why the virus is so deadly. The mortality rates can reach up to 90% depending on the virus species. The current outbreak in Liberia, Sierra Leone, and Guinea has killed approximately 380 people so far. When I presented at my poster session, this number was about 1,000. And so as you can see, this outbreak is really starting to gain momentum. And as a result, this created an increased demand for diagnostic tools, therapeutics, and all of a sudden there's just a lot of money going into this research right now. And so the present study involved testing the secretory glycoprotein associated with all 5 known Ebola virus species. When I originally started, I thought that these were all different strains of Ebola, but it turns out all of these are genetically and so antigenically distinct, they were actually classified as different species. And then among these, there's also strains such as Zaire Gabon strain, Zaire Mainga strain, so on and so forth. And this present study sought to create a new antibody panel that can detect the virus in human serum. And so the antigen in question, as I've already stated, was the Ebola virus secretory glycoprotein. The glycoprotein of Ebola is used to promote cell entry, and it consists of two subunits called GP1 and GP2. The GP1 subunit, as it approaches the cell membrane, is cleaved by a protease called furin. And these two subunits are held together by what's called a disulfide bond. You're going to learn more about this in biochemistry. But anyways, that bond is cleaved, and then GP1 associates with the cell membrane to identify a particular receptor. GP2 actually penetrates the membrane and promotes the entry of the virus into human cells. But while this is occurring, it also releases copious amounts of immature glycoprotein into the blood. And this is currently hypothesized to serve as a decoy for the immune system. There is evidence that it can also interfere with the function of neutrophils, which also can contribute to hemorrhagic fever and the cytopathic effect that can be observed in infected cells. And in order to make this antigen, I mean, naturally, I couldn't work with wild-type Ebola virus in the lab I was working in. Yeah, I'm really grateful for that. I value my life.
Good.
But anyways, we used, well, it's an embryonic cell line called 293 T cells. We're actually transfected with expression plasmids. And how these assays work are, you have the plasmid, and these plasmids typically encode the protein of interest, but then they also encode for a histidine tag that can be used in a form of affinity chromatography, and that just makes purification a little bit easier. And it has to be combined with a transfection reagent, which basically allows the plasmid to enter the cell, usually by endocytosis, but depending on the assay, this can vary somewhat. And then after the 293 T cells were transfected, they were cultivated for a couple of days. The media that we used was called Delvecco's modified eagle media, and it's basically modified with fetal bovine serum. It's really expensive stuff. It costs about $400 for half a liter. And so the grants needed to do this research are really quite large. And so the secretory glycoprotein was then collected by centrifuging the cells, collecting the supernatants. And then they were exposed to magnetic nickel beads that actually have a high binding affinity to the histidine tag. And then all of the other proteins found in the bovine serum could just be washed away. And then we would just have pure antigen. In this case, pure secretory glycoprotein. To create the antibodies, this is overall a very difficult process. The cells used to create these are called hybridomas. And these hybridomas are created by vaccinating mice against a particular antigen. In this case, it was the secretory glycoprotein produced by the Mainga strain of the Zaire species. And basically, how it works is the mice are vaccinated in intervals. B cells are then extracted from the spleen. And then from there, these B cells are then exposed to myeloma cells. And under a certain set of conditions, these cells will fuse. And so we basically have a cancerous cell line that continuously produces a certain antibody. And then these antibodies can then be collected, and then from there they can be further characterized in the lab. In this lab, we used an assay called an ELISA, which I'm going to discuss in a little bit more depth here in a minute. But what I can tell you is that this kind of research here, this will get you pretty much on the enemies list of all PETA and Greenpeace people. Yeah, they're not too fond of us and the things we do in our lab. But from there, we're able to conduct an enzyme-linked immunosorbent assay. And to conduct these assays, we have a specialized plate. And then from there, we take our antigen, we just put it in the plate, we put it in the fridge overnight, and then generally the antigen will bind to the plate very effectively. And then from there, we take the antibody panel and we expose them to the antigen, and then we incubate them. And then after that, we have to treat the experimental antibodies with a secondary antibody that's capable of binding to a particular substrate that'll promote absorbance. For your substrate, you can use a chemical called PNPP, horseradish peroxidase, streptavidin. When exposed to the secondary antibody, all of these substrates will create basically immunofluorescence, and then from there, this immunofluorescence can be measured quantitatively, and this will give you an idea of how well the antibody is capable of binding to the antigen. And then from there, we're able to collect these curves, and the first antibody in the panel was called S3. And this is an antibody that's used solely for detection. And we discovered here that this antibody has a relatively low binding affinity, but it still is capable of detecting the Zaire strain, the Ivory Coast strain, well, not strain, species, the Sudan strain and the Reston species. Actually, no, I'm sorry. They can detect Zaire, Sudan, Reston, and Bundibugyo, but it's actually not capable of identifying the Ivory Coast species. The S9 antibody that I worked with is actually the most interesting because it can be used to identify the viral antigen, but it can also neutralize the virus in vitro. And this actually could provide a basis for creating a vaccine. And part of my goals here was to purify the antibodies and actually send a sample of this one to the Scripps Research Institute. And then from there, they could do X-ray crystallography to create a crystal structure. And then from there, we can determine exactly how the antibody binds to the antigen. And this could create a basis for a vaccine or for a detection assay. But we discovered here that it's actually capable of detecting the Zaire species exceptionally well, which is the species that's been implicated in the current outbreak. And it can also detect the Ivory Coast strain moderately well. However, it's not capable of detecting Sudan, Reston, or Bundibugyo really at all. The S17 antibody is a broadly reactive antibody, and we discovered that this is capable of detecting all known Ebola virus species. And the S12 antibody had many of the same characteristics. We discovered that it can detect Zaire, Ivory Coast, Sudan, Reston, and Bundibugyo. And if we can characterize these two antibodies, this would actually could create a basis for a broadly protective therapeutic if we can learn what particular epitope or what part of the antigen these antibodies bind. And then after I conducted the ELISAs, we decided to do an immunofluorescent assay. And the protocol is basically very similar to what's done in an ELISA. We did infect, well, these were all cells that were infected with wild-type Ebola virus species. And then from there, they were able to be taken up to the biosafety level 2 lab where I was, and then I was able to conduct the assay. But then we transfected actually Vero E6 cells. They're monkey epithelial cells. And then as a positive control, we transfected these with, I'm trying to think, GFP. It's basically a immunofluorescent protein. And then we have a negative control, which are basically cells that were treated with DAPI, which is a nuclei stain, just to establish the fact that we actually have cells there. The DAPI images aren't included, but this is to show that the negative controls didn't produce an excessive amount of background. And then overall, the Zaire Gabon strain didn't provide very promising results. Interestingly, the S9 antibody was able to detect the Zaire Mainga strain after the glycoprotein has been incorporated into the cell membrane. And then unfortunately with Reston and Sudan, we actually were able to obtain also negative results. But the point of this assay is basically to show whether or not virus-infected cells can be detected. And in this case, it seems that the glycoprotein, after it enters the cell, it undergoes a conformational change. And it seems that, well, with most of the species, the antibodies lose their binding affinity to the epitope for reasons that we were not able to fully understand. And so, in conclusion, results were successfully obtained. Unfortunately, we did have some problems with purifying the antibodies, which means basically separating them from the bovine serum proteins. And that's basically the bulk of the research. I did do other miscellaneous projects, but overall this was a pretty successful study, and it looks like it actually will result in a publication that'll be coming out in April. And so it was pretty promising.
Okay. Do anything.
Yeah, you gotta get rid of your face, Karen. Sorry.
Okay. My name is Karen Chanchak, and what I researched this summer was the characterization of fugitive peroxisomal proteins in Arabidopsis cells. So, some background on what peroxisomes are. They are small, nuclear membrane-bound organelles found in eukaryotic cells, and they're responsible for a lot of metabolic functions. For example, the catabolism of long-chain fatty acids for metabolic purposes, and the metabolism of hydrogen peroxide to deactivate or remove the oxygen radicals. There are many peroxisomal disorders found in mammals, including mice and humans, which result in early death. So an example of one is Zellweger syndrome. Usually affects infants, and they don't live longer than a year or so in life. So unlike mitochondria or chloroplasts, peroxisomes lack their own genome. Their genes are nuclear oriented. Their proteins are synthesized in the cytosol and then transported into the peroxisome. And the way that happens is they have to have a PTS1 or PTS2 signal, which is a polypeptide signal. And there's here this part and the highlighted, that's PTS1, and then this is PTS2. They have to have that signal to come into the peroxisome. I worked with Arabidopsis thaliacea. That's a great picture for you guys to see. It's a flowering plant in the mustard seed family. It has a sequenced genome, which means that we know the genes, but we don't know what all of them do. It's transformable. The gene expression can be modified through tDNA insertion and RNA interference. It's a weed, so it's reasonably fast growing. It's small, so it's great for labs with not a lot of space, and it's easier to work with. A lot of eukaryotic cells, as I mentioned before, have peroxisomes, and so Arabidopsis have peroxisomes that are built and function in a similar manner as found in mammals. So it's useful for biomedical research. So the question I had this summer was, do the proteins encoded by ATAJW1 and ATAJW12 localize to the peroxisome? These are not the formal names, we rename them, but we'll get to that later. I hypothesized that the proteins may localize to the peroxisome because of the putative PTS1 signals that we found in the sequence of the genes. So the proteins are captured, PTS1 proteins are captured by the peroxisomes in the cytoplasm, and then the protein receptor unit is transmitted via protein complex into the peroxisomal matrix. The signal peptide is cleaved off, the protein is folded into a functional state, and then the receptor is recycled back into the cytoplasm. So these are the formal names of the genes I was working with this summer, but like I said, we renamed them because they are unknown. They're around the same size. Of course, you can see ATAJW1 is larger than ATAJW12, and both contain the putative signal that encodes for being brought into the peroxisomal matrix. As I said, function are known for both genes, and peroxisomal location has not been determined. So here's an example of my cloning efforts for my genes of interest. So we clone from available stocks. We have a record of 1,000 base pairs, which ATAJW12 is supposed to be around 750, and ATAJW1 around 790. And you can see the first 6 lanes are around 750, and then they go up around 790. So that was successful. We then put our genes of interest into a sequencing vector, and which our vector was around 4,000 base pairs. And again, you can see our vector here, and this is ATAJW12 around 750. And I did it for both genes, but I'm just showing you guys the information for one gene just for time. And then we had them put into an expression vector, EYFP. Now the YFP on this up here stands for yellow fluorescent protein. So it actually glows yellow when you look at it under a microscope. So again, our base pairs, 10,000 and 1,000, and our proximal expression vector is around 10,000, so 11,000. So you can see right here, and then the ATAJW12 around 750. So again, we had them work, and so we moved on. So this whole slide right here is really awesome because Dr. Ryman never could get this far. Well, he got this far, but never could go further than this. So thanks to these cool hands here and really awesome music for numerous amount of hours, we could get past this. So anyway, that was a great moment of my life. So we're going to be doing implants of peroxisomal localization for both genes. So we're going to infect the tobacco plants with agrotumophagians, which is a natural bacteria that infects, which will be carrying our genes of interest with our vector, EYFP. And again, like I said, we will infect the tobacco plants. The Olsen lab at U of M will be using their confocal microscope to see if they see fluorescence from our genes of interest from the peroxisome. So like I said, EYFP, the yellow fluorescent protein, you will see a yellow light emitted, so kind of like this, from the coming from the peroxisome, meaning that, yeah, they're in our proteins are in the peroxisome. So here's our gene of interest in protein expression vector. around 5,400, again, vector, and then for ATAJW1 is on top and ATAJW12 is on bottom. So again, around the same, they came out. Basically, we want to characterize the function and structure of the proteins, and to do that we need to make the protein, and this is one of the first steps to doing that. In addition to all that I just talked about, I also did a tDNA in vivo impacts and catalyst assay. And for the first side project I did, I looked at the growth and development of two different, of the same, of Arabidopsis, but I looked at the mutant and wild type, just for growing differences. And I identified tDNA homozygous plants and in my mutant plants. And I did enhance the genomic DNA extraction that we will be using in a lab this fall. I also did a catalyst assay to determine the activity in lettuce and Arabidopsis. We treated the plants for 3 weeks with ammonium chloride and determined if there was any difference between the activity in a wild type and a mutant. The data was inconclusive, so we will still be working on this some more. So, in conclusion, I cloned out my genes of interest into 3 distinct vectors, and we are currently waiting on the implanta expression analysis from U of M of my genes, and we're in the process of characterizing the structure and function of my recombinant protein. And like I said, we're going to try and continue to work on the Catalyst project to make it a functional tool for classrooms. So, acknowledge it. I'd like to thank Dr. Wyman for this research opportunity, Mr. Castle for providing some slides and for conversations on discussions of just proximal stuff and really other stuff. Allie for leading the efforts of my implanta expression localization. Dr. Laura Olson and the Olson Lab for providing necessary research tools and for other great things. And Springer University Departments of Biochemistry, Biology, and Chemistry for equipment and supplies provided this summer and for general support, and the McKenna family for financial assistance.
So, if you have any questions, please ask them. You can ask of any one of the presenters. And then when you give your response, hold the mic and touch your chin, please. Dr. Baratovich, what a surprise.
John,
Yeah.
the mutant that you made that had a lysine at the active site,
Yes.
can you if you leave the arginine at the active site, can you make any adducts with the arginine? With any, you know of any substrates that you can use that'll make an adduct with the arginine?
Oh, the inhibitor?
Yeah.
Yeah. We didn't investigate that. That there's potential. I think we swapped for the lysine based off of a paper done by another MSU PI who had used, I think, retinal with an enzyme, swapped arginine for lysine, and was able to produce this very colorful colored enzyme. And so the reason we did that is based off of what he did, and I believe when they tried it, the arginine didn't work as well as lysine, so that's why we switched for lysine. And for those of you that aren't familiar as much with amino acids, the lysine is pretty similar. It just has the one amino group at the end versus 2 for arginine. Any other questions? Yeah.
Mine's a logistic question. When did you start looking for these REUs? When did you apply? And how many did you apply for?
Yeah, so I was looking, my freshman year, I wanted to get into one. I applied to only 2 or 3 and didn't get into any. So the following year, my sophomore year, I applied to, I believe, 5 different REUs, and I looked for programs around September, October, worked really hard on developing like a resume, a cover letter, talking to professors for references, things like that, personal statements. Each one requires a little bit different kind of format, but then I sent in my applications at the end of December, kind of early January, I'd say. The deadlines aren't usually until around February for these RU experiences, but if I tried to get mine in just a little bit early before that deadline, just to make sure everything was in okay. Yeah.
Great.
Okay, if I remember correctly, I think I started applying around December or January. Usually the deadlines for these programs are February at the very earliest. A lot of them will set their deadlines for around March. It's just that in order to really be ahead of the curve, you need to confront professors fairly early just to ensure that they can write you a proper letter of recommendation. And at least in my experience, you're not going to get in everywhere you apply, but you know, if you find a few programs that you're really, really interested in, chances are you'll get into at least one. I mean, in my case, I got into UTMB and I got into Emory University. There were a few other places I applied to. For instance, I'm trying to remember off the top of my head, the University of California at San Francisco. I applied from there and I got rejected. I got rejected from the University of Nebraska, from U of M, and a handful of others, but you know, if you apply to enough of them that you're genuinely interested in, and if you can write a personal statement that shows that you're genuinely interested in the research that they do there, then chances are you will find a good fit.
I have something to add real quick. If you apply, I highly recommend that you look at the labs that are participating in the RU and investigate what research that they're doing, and then state that in your application that you're interested in so-and-so's research because X, Y, Z, like how you are interested. So, any other questions? Yeah.
Jeff? So the antibodies which were made in an infected critter didn't bind to the actual antigen when it's presented on the cell surface. Do you think that's because the antibodies were largely made against decoy glycoproteins?
It could have been that. I wasn't actually the one who made the hybridomas. He had an intern before me who made them, and I just took over his research. I discussed why this could be with a postdoc in an adjacent lab, and he actually did his PhD on the cross-reactivity of antibodies basically against the flu for a flu vaccine. And he said that usually when you do these ELISAs, the proteins can assume unnatural conformations. Like for instance, the antibody that he worked with is usually in the shape of a protein. But then after the virus gets incorporated into a cell, it undergoes a conformational change, and I think he said that it kind of assumes a more linear orientation. And so after the virus enters the cell, it just seems like it's, you know, undergoing a conformational change that, you know, it seems to be blocking the epitope.
I wonder if you should be coating your wells with cells instead of actual protein then. To assay the affinity and avidity of your antibodies.
That probably would have been the next step. Yeah, but it was, you know, I ran out of time. That would have been a nice side project, though.
I think we have time for maybe one more question, if there is any. If not, yeah.
On one of your slides, there was a squiggle line on the beta amino acid.
Yes.
What in the world is about that squiggle line, John?
So If you recall that table of the previously studied immunomutases, they had various different stereochemistries, and so the squiggle line represents that we're unsure what stereochemistry it exactly has. So, for
So
example,
it could be what two choices?
it could be an R or an S stereochemistry.
Oh, that's what a squiggle line means.
Yeah.
Organickers are listening to this, yes?
So I chose to represent that in that slide because it could be either or depending on which particular immunomutase you use.
Thank you.
Yeah. All right. Thank you.